Hello, my name is Andres Vidal Gadea, and I'm a molecular neuropathologist interested in the molecular and cellular basis of behavior. Our lab uses multidisciplinary approaches, including molecular, robotic, optogenetics, and in-depth behavioral techniques. We are funded by the NSF to study the molecular and cellular basis of magnetic transduction, and we have received NIH funding to study the pathophysiology of Duchenne's muscular dystrophy. Our students regularly earn awards for their work and publish in high-impact journals such as eLife and PNAS. Funding is provided through a combination of research and teaching assistantships. We are looking for graduate students to join both of our projects in the fall of 2021. Thank you.
I'm a neurophysiologist, and my lab studies what makes neuronal activity robust against perturbations, such as changing temperature conditions. We know very little about how global warming affects the nervous systems of billions of animals and why fever endangers the human brain. We explore temperature effects by focusing on neuromodulators in crab and crayfish species to extract evolutionary and general mechanisms of neuronal temperature responses. The large crustacean neurons and their known connectome and neuromodulators make them ideal for electrophysiology and molecular experiments to identify the underpinnings of temperature effects. As a grad student in my lab, you'll join an exciting research team and experience direct mentorship from me while you work independently on your project. To prepare for your future career, you'll publish papers, submit grant applications, and present at international conferences.
Hello, my name is Paul Garris, and my laboratory studies dopamine neurons. We investigate the mechanism of psychostimulant action with the focus on amphetamine. We also collaborate with engineers at Case Western Reserve University and University of Michigan to advance dopamine sensing technology.
Hi, I'm Alicia Varales Mortimer, and my research focus is on understanding how and why we age, and looking at the interaction between genes that regulate aging and environmental factors, such as pesticides and heavy metals, contribute to diseases of aging. And my lab, we use fruit flies, Drosophila melanogaster, as our study system. And that's because flies are surprisingly similar to humans. About 75% of all disease-causing genes in humans have an equivalent in the fly. And when we mutate these genes in the fly, we give them the fly version of that disease. And that really helps us understand what are the molecular mechanisms that might be driving disease processes. And I'm interested in both muscular dystrophies as well as several different neurodegenerative diseases, including Charcot-Marie-Tooth disease and Parkinson's disease. My students have all been authors on different publications, and I routinely take my students to both regional and international meetings. So if you are interested in studying relationships between genes, environment, and how this affects aging and age-related diseases, please check out my lab.
Hello, my name is Ryan Paitz, and I'm an assistant professor of endocrinology here at Illinois State University. I also happen to be a product of Illinois State University. I earned my PhD here in 2010, so I can speak to you firsthand about how Illinois State's a great place for grad school. I think one of the things that sets us apart as a university is being a little smaller. Whether you're a graduate or a master's student or a PhD student, you're going to get the opportunity to directly interact with your advisor. You're not going to get lost in a giant lab full of postdocs and senior graduate students, and I think that's something I take very seriously in my lab, that direct interaction with the students, where we study how developing vertebrate embryos regulate their exposure to maternal steroids during development.
I'm Martin Engelke, and I joined the faculty at Illinois State University recently. In my lab, we are interested in how molecular motor proteins function and how they're regulated. The undergraduate and graduate students in my lab use various tissue culture techniques combined with advanced fluorescent microscopy, CRISPR-Cas9 mediated genome editing, and computational analysis to address these questions. I established my lab a year ago, and we are currently growing, so we are looking for highly motivated graduate students like you. Growth is facilitated through external funding, which I recently obtained, and the contribution of motivated and talented students. This is why you can expect excellent mentoring in my lab. In addition to me, my lab manager, who holds a PhD in neuroscience, is also heavily involved with training students. I am looking forward to your application.